Thank you all for being here. Today's topic of our Lunch and Learn is investigating water quality in the complex ecosystem of southern San Francisco Bay. And we were talking sort of before this meeting started, a topic, water quality, nutrients, not as photogenic as wildlife, or as like above the fold in the popular press as things like sea level rise and things, but it's super important and interesting and complicated. It just doesn't photograph as well. San Francisco's Lower South Bay connects to a very complex network of sloughs and marshes and former salt ponds that are in various stages of restoration and enhancement. The water moving in and out of those ponds, coming from wastewater treatment plants and other places, runoff, moves around, and it creates a very complicated set of interactions between all of those different habitat types. And it's really important for us to know about these sloughs and the ponds and these shallow waters because we're doing work in them and we're trying to avoid creating conditions that might worsen or complicate or have any adverse effects on fish, on wildlife, on vegetation, on people. And so it's just a really complicated topic that we need to better understand. And it ties into the project's adaptive management plan, which was set forth over 20 years ago. Behind the scientific question, what is the effect of restoration project pond management and increased tidal prism from tidal marsh restoration on water quality in the South Bay? And to address that question is today's speaker, Ariella Chelsky, excuse me. In this talk today, Ariella will explain what we have learned about these complex interactions using data from water quality sensors and complex model analysis. She's a managing senior scientist at the San Francisco Estuary Institute, where she oversees nutrient biogeochemistry and monitoring within the nutrient management strategy. That NMS, nutrient management strategy, is a research program that studies the impacts of nutrient loads on San Francisco Bay and evaluates the potential management actions that can be taken in response to those. Ariella has a Bachelor of Science in Marine Biology from the University of British Columbia in Vancouver, Canada, and a PhD in Marine Ecology and Biogeochemistry from Griffith University in Australia. She's also did a postdoctoral fellowship at Louisiana University's Marine Consortium, where she studied the impacts of the Deepwater Horizon oil spill, remember that, on salt marsh biogeochemistry and plant ecology. So we're grateful for her time with us today. We're thinking the talk will be about 30 or 40 minutes, which will be plenty of time for Q&A at the end. So please do prepare some questions, which you can ask either vocally after her presentation, or you can put them in the chat box or Q&A box at any time. I'll be sort of trying to facilitate the Q&A at the end and grouping questions as we go. So with that, I will stop talking. That's our opening credits. And here's Ariella Chelsky. Thanks.
Thanks, Dave. Thanks for the intro. Yeah, so as Dave mentioned, I work on the nutrient management strategy. And so we're studying the impacts of nutrient loading on water quality in the Bay. And along that vein, this talk will be focusing on nutrients and water quality in the Lower South Bay, specifically looking at the former salt ponds and sloughs and the interactions between the two. And for this talk, I'm presenting work from a couple of different studies. On this title slide, I'm listing the primary co-authors. Martin Valerich and Derek Roberts led most of the work that I'm presenting, but there are a lot of other collaborators and contributors who have listed here. These are people who helped with the data collection, data analysis, and modeling. And I'm sure this doesn't capture everyone who's participated, but these are kind of the main contributors to the work that I'm presenting. And also we have the funding sources here. So just to orient people, this is a map of the South and Lower South San Francisco Bay. I think most people joining are familiar with San Francisco Bay, but yeah, this is a Golden Gate here. It's where the Bay Bridge is. And for this talk, I'm going to be entirely focusing on the Lower South Bay in this pink box. And it's a really complex system, as you can see from the bathymetry. And Dave touched on this as well in the intro, but there's a lot going on in the Lower South Bay. So there's this network of shallow sloughs. There are managed former salt ponds that are in the process of being restored. There's water flowing between the bay, the sloughs, these restored tidal areas, and managed ponds that are creating complex interactions between the different habitats. And adding to this complexity is the fact that the Lower South Bay receives treated wastewater effluent from three wastewater treatment plants that service the densely populated surrounding regions. So these three discharges are San Jose, Sunnyvale, and Palo Alto. And they nitrify their effluent, and loading has been going down due to upgrades in the treatment process, but they still have really large nitrogen inputs to the system. And here's a couple of figures to illustrate this point. On the left, this is DIN, or dissolved inorganic nitrogen, loading to the Lower South Bay from the three treatment plants. So the different color bars represent the different dischargers, with the San Jose, the reddish pink bars being the largest discharger to the region. And the data here from about 30 years. You can see over this time period, there were significant reductions in nutrient loads over this time period. And this is from a report, an SFEI report from 2016. So it is kind of only covering up until, yes, like until 2011. So it's kind of cut off in the more recent years, but loads have continued around 8,000 kilograms of dissolved inorganic nitrogen per day each year. So, and then on the right is a figure showing dissolved inorganic nitrogen concentrations in different sub-embayments in San Francisco. And these data are from the USGS Peterson cruises that sample the channel of the bay every month. And this program has been going on for decades. And despite this effort to reduce nutrient loading, the Lower South Bay still has the highest nutrient concentrations of any sub-embayment in San Francisco Bay. So yeah, this is Lower South Bay on the rightmost side, and yeah, has the highest concentrations. So what are these nutrient concentrations, nutrient loads and concentrations mean for water quality? Well, this is a conceptual diagram showing the effect nutrients have on coastal environments. Essentially what happens is that nutrient inputs enter coastal systems. It can be from many different sources, including agriculture and stormwater. In the Lower South Bay, most nutrients are coming from the wastewater dischargers. So they enter the system. And then the nutrients in these coastal systems can then stimulate algal growth or phytoplankton growth in the water column. So it kind of fuels this growth of algae or phytoplankton. And then once that growth kind of dies and sinks down and starts to decay on the seafloor, the bacteria use oxygen to break down the algae or phytoplankton. So this decay or decomposition process is also known as respiration. That draws down oxygen. So that creates these like low oxygen areas. And that means that there is then less oxygen available for biota like fish in the water column. And this process depends on nutrient levels. It doesn't necessarily mean just having nutrients in the system doesn't necessarily mean that oxygen is going to drop low. It's dependent on many different factors, including the movement of water in the system. But if nutrient inputs are really high, it can lead to oxygen problems. It can lead to low oxygen. So to see if oxygen concentrations are actually low in the Lower South Bay, I have this plot here that shows oxygen concentrations at in several Lower South Bay sloughs. This figure is showing time on the x-axis, so it's going from 2015 to about 2023. And then on the y-axis is dissolved oxygen concentrations in milligrams per liter. The color of the figure is also showing oxygen in milligrams per liter, so it's kind of helpful to differentiate when oxygen concentrations are low or high on the graph. And then each panel is a different location in the Lower South Bay. So the four bottom panels are all different sloughs. And then the top panel is a channel station or a location in the channel at Dumbarton Bridge. And so you can see that oxygen remains pretty high at the channel station. We don't frequently see these dips where it's turning where the oxygen concentration is kind of like in the red zone, so that anything that's a yellow to red is 5 milligrams per liter or lower. And the reason I'm using that cutoff is the San Francisco Basin Plan. The oxygen threshold for the bay is 5 milligrams per liter. So that's not actually the threshold that applies to the Lower South Bay sloughs, but it is a good kind of like threshold or like a point of reference when looking at these data. And you can see that in a lot of the Lower South Bay sloughs, especially in Guadalupe and Alviso Slough, these two middle panels here, these concentrations of low dissolved oxygen are pretty frequent. So this is happening fairly frequently in these sloughs. So I guess the question then is, who cares? Does this matter? Do these these low oxygen levels have any negative impacts? And it's the fish and other biota that care. So there are a lot of fish that use Lower South Bay sloughs as habitat. I screenshotted this cover. This is a cover art from a report that Levi Lewis wrote for SFEI, and I love the artwork on this because it shows all like the many different fish species that are found in the Lower South Bay sloughs, and this includes species like sturgeon, longfin smelt, anchovies, and shad. And there's also other organisms that are living in these sloughs and rely on the oxygen levels in them, and that include things like crabs and clams. And so there are a lot of fish that are found in this part of the system, and they need oxygen to survive. So that's why oxygen matters. Whether or not these oxygen levels are high enough to sustain the fish populations that are found there has been the subject of a study that we've been doing at SFEI in collaboration with the Regional Water Board, Tetra Tech, Levi, and a few other researchers as well who study the effects of low oxygen on fish. And just based on some of the analysis, we found that based on the oxygen requirement for fish, that oxygen is dropping low enough in some sloughs to be problematic for sensitive species. So we can say the data is telling us that from these other analyses that these oxygen levels may be problematic for the fish in the system. There are a few potential causes of low dissolved oxygen in the sloughs. So I've already explained the linkage to nutrients, to high nutrient loads. But another kind of player in this part of the system is the managed ponds that are connected to the sloughs. We think that they may also be playing a role. They have a really massive footprint in the Lower South Bay. So it's possible, very possible that they are impacting water quality in the sloughs. And that's kind of the point of the work I'm presenting today and what we want to investigate. So we have a few different questions about these interactions. So far, our work has focused on this first question: how does the interaction between high nutrients and the former salt ponds impact water quality in the sloughs? An important I guess, thing to mention here is that the former salt ponds are not all the same. This is something that Dave Halsing, a point that he's made many times in meetings with me. Their hydrology is quite different, and they're in different stages of the restoration process. So they're not all behaving the same. This question and our analysis is really focused on former salt ponds with limited exchange with the bay that are not restored yet to wetlands. So we use Pond A8 complex as a model for ponds with limited exchange. And this has been a focus for a lot of our work because it's connected to sloughs with the lowest oxygen. So I'm really today going to be focusing a lot on Pond A8 and Alviso Slough, which I'll show in a figure in a minute. And the other questions that have kind of come up from this work as well are: how will water quality be affected by planned restoration activities? And can restored ponds help buffer nutrient inputs from nearby wastewater treatment plants? So this is not, these are questions that we have, but this hasn't been the focus of our analysis so far. So I'll just talk about them very briefly at the end, but I don't have answers to them, unfortunately. So our hypothesis is that for the combined effect of ponds and nutrients on slough water quality is shown in the schematic here. So we think that nutrients are delivered from the ponds via, delivered to the ponds via tidal transport. And then this nutrient-rich water fuels high biomass production in the ponds. And these are, they're large, they're shallow. There's a lot of light that's reaching the water, so they're kind of perfect conditions for phytoplankton to grow. And they also have longer residence times because of the limited exchange with the bay. So it's giving algae a lot of and phytoplankton, it's giving phytoplankton a lot of the fuel needed to grow and then time for them to develop. And we think then that the biomass or phytoplankton is exported back into the sloughs where degradation and can lead to high respiration rates and low DO. I'm not sure. I guess there are maybe people from the general public who aren't as familiar with these processes. So I thought it would be helpful to go back to this conceptual model or this diagram and kind of explain how things are happening using this diagram. Essentially, we think what's happening is what's in this conceptual model, except that there's a spatial decoupling. So what we think is happening is that the wastewater treatment plants are delivering the nutrients to the sloughs, and then that's being advected into the ponds. Then the managed where the algae is growing in the managed ponds, and then the decay process is happening in the sloughs. So we think this is all happening, but just kind of like spatially decoupled. And so to test this hypothesis, we used a combination of fixation data, analysis, and synthesis and modeling. So I'll talk through all of those. And most of the data we use for this analysis come from the nutrient management strategies monitoring stations. We actually monitor water quality all throughout Lower South Bay, and Central Bay. But I'm just showing our stations in the Lower South Bay. And these are moored sensors or fixed stations that have exosondes. These are water quality sondes that are measuring water quality parameters at 15-minute increments. And they measure things like dissolved oxygen, chlorophyll, temperature, and salinity, among other parameters as well. And a couple of these stations were established in 2013. So they've been there for more than 10 years. And then we've added several stations throughout the years as well. And then the two blue ones here are retired stations, but we still have several years of data at both of those stations. And so today I'm going to be focusing on Guadalupe and Alviso Slough, and I'll also show some of the data from the pond stations as well. We collaborated with Stillwater Sciences and Valley Water to add 2 sensors to ponds in Lower South Bay over the last year. So I'll be kind of focusing on that part of the system. So we have some preliminary data from these stations to support the proposed mechanism. So this is kind of, this is some evidence that we saw in the water quality data kind of in the early stages that is kind of what led to us developing the mechanism, the proposed mechanism. These are just based on a single time point corresponding to gate operation. So just some preliminary data. And what we have 2 examples. So this one is showing chlorophyll data at our Alviso station when gate to pond 8 it was closed and then reopened again. So you can see when the gate was reopened that there is this pulse of chlorophyll coming out of the pond at our Alviso station, or that's what appears to be happening. And we saw a similar but much more dramatic pattern at our Guadalupe station when gate for pond A3W was opened. Again, you can see this kind of dramatic increase in chlorophyll at that station when the gate to the connection to the pond was reopened. So this kind of supports the idea that chlorophyll is maybe growing in the ponds and then being evected out into the sloughs. We also have some high-speed mapping data that the USGS collected on an ebb and a flood tide. This is really cool. We've done a lot of work with them in the South Bay, and they do a lot of work in the North Bay as well. What they do basically is they zoom around their boat, they pump surface water up and run it past a bunch of different instruments that they have on the boat. So they measure all these parameters as they're driving around. And so you get this really cool map of what's happening as like in a single snapshot in time. So you really, it helps you kind of understand like what's happening spatially within a system. And in these figures at the top panel is showing dissolved oxygen concentrations. And with like the red or maroon indicating lower concentrations and the blue colors indicating like higher DO levels. Then the green is chlorophyll. The yellow to pinky purple is nitrate. And on the left is the data from the flood tide. On the right is the data from the ebb tide. And on the flood tide, what we can see is that when water's coming in from the open bay, DO is high. So there's like this oxygenated water coming in from the open bay. Chlorophyll is low generally, except for like a small region in Alviso. And nitrate is intermediate. It's kind of variable depending on which slough they were mapping. But then on the ebb tide, we see as water's coming back out from the sloughs and the ponds, oxygen is much lower. Chlorophyll is a lot higher, especially in Alviso. And nitrate in the sloughs with the connections to the wastewater treatment plants are a lot higher, but it's actually lower in Alviso. So what it looks like is that nitrate is going in on the flood tide here, and then being delivered to like down up slough and to the ponds, and then on the ebb tide, low oxygen, high chlorophyll are coming out. And then I guess one more piece of preliminary evidence, and then I'll get to some of the other study results. But in 2023, as I mentioned, we collaborated with Stillwater Sciences and Valley Water to add sensors to pond A4 and A8. They were deployed for about a year, and now the study is completed, but we're hoping that we can at least continue some of the monitoring in pond A8. And what we saw in the data is that there are periods of really high chlorophyll in the ponds. This is just an example from a snapshot from pond A8 from November or September and November last year. And in this plot, I think it's a little hard to see, but the chlorophyll is essentially going up to about 600 micrograms per liter, and it's staying sustained at that level for quite some time. And this is not 600 micrograms per liter is outside of the range of what the EXO is calibrated to measure. So it's not precise, but we can say that it's very, very high. This is this like the amount of fluorescence that's happening for the EXO to be reading levels like this means that there's a lot of production, a lot of chlorophyll in the ponds. And just to put this in perspective, this is much higher than chlorophyll levels in the bay that we measured during the harmful algal bloom that happened in 2022. So just to put that in perspective for you, they're very, very high. And even when chlorophyll is low, kind of like in some of these areas that look like it's a little bit lower, it's still actually quite high because it's around 50 or 100 micrograms per liter. So that's kind of supporting this idea that there is a lot of chlorophyll production happening in the ponds. So like I said, these plots are just showing observations. They're not telling us a lot about the mechanism. So to dive deeper into these patterns, we went beyond the observational data and used moored sensors to calculate rates of oxygen uptake or respiration in the sloughs. This is work that was led by Derek Roberts. And there are two primary stations, Newark Slough and Alviso. Newark Slough extends from the northern edge of Lower South Bay into broad tidal marsh. It's separated from active commercial salt ponds by levees. And although it was part of the study, it's not a big focus here. I'll just show it as kind of as a point of comparison for the Alviso data. So the Alviso Slough is the primary focus here. It has several connections to ponds that have been restored to tidal action via gated weirs and levee breaches. So this is our example station where it's a slough that has pond connections. And we also use data from the pond A8 outlet, which is positioned right at the pond connection between Alviso and pond A8, sometimes referred to as the notch. So it's right at the where the gate opens and the water from the pond is coming out into Alviso Slough. And Derek, for these calculations, he used these high frequency sensors to calculate rates of respiration or oxygen uptake. And how this works is that you have the sonde and you have a parcel of water passing by. So it's measuring dissolved oxygen concentration on the flood tide. And then on the ebb tide, the same parcel of water comes back past the sensor. And what he did is he calculated the difference in oxygen concentration between the flood and the ebb tide in order to estimate rates of respiration or oxygen uptake. This is based on 5 years of data from 2015 to 2019. So there was about 1,000 tidal cycles per each site to use for the calculation. And going back to this handy conceptual model, we were just to put this in perspective here, what we were trying to estimate is this process in the circle here. We were trying to estimate the decay of algae or phytoplankton that's happening in the sloughs and the associated drawdown of oxygen. I guess it's important to note that we were only using data collected at night. So that's why we were only measuring respiration. We weren't measuring any algal growth or production in the oxygen signal. So first I'll talk about the dissolved oxygen concentrations, and then I'll dive into the respiration rates. So this figure shows dissolved oxygen and chlorophyll at both stations over the 5 years of the study. So the panel on the top is dissolved oxygen, and the panel at the bottom is chlorophyll. And the orange line is Newark, and the kind of green blue line is Alviso. And this is showing seven-day averages in 7-day moving averages in concentration. So it's not capturing like all the variability that's happening, but it's showing you the average pattern. And the main takeaway here is the seasonality. So dissolved oxygen levels are generally peak in winter and early spring, and then DO minima occurred in spring or summer, as we expect when temperatures are higher. And then chlorophyll peaks at Alviso were coinciding with the DO maximum in the winter and early spring. And there was some variability with the Newark chlorophyll data, but it doesn't show quite the same seasonality as the Alviso station. So when we look at respiration rates, these are rates averaged by month for the 5 years of the study. So each bar is showing a different month throughout the year. And so we're using these plots to show you the seasonality in the patterns and how they differ between the sites. So we have Newark on the left and Alviso on the right. And they're showing, well, actually, there are bars at the bottom that are showing production, but I tried to block those out from the figure caption or the legend, because I'm not focusing on those data for this talk. But what we see here is that there are really different seasonal patterns between the two sites. So at Newark, we see these strongly seasonal rates with the highest respiration rates occurring in the summertime. This is what we generally expect, that when there's warmer temperature, that's usually associated with higher rates. We think there'd be more production and more respiration happening. But this is not the pattern we're seeing at Alviso. It's a little more complicated. And what we see at Alviso is that the highest respiration rates are happening in the winter and the spring, and then respiration decreases into the summer. So in the summertime, when we think rates would be highest, they're actually lowest. And what's interesting here is that the peak in respiration doesn't line up with the seasonal patterns in dissolved oxygen. And so this plot at the bottom is just a one time span, like 1 year of data from one of the previous plots, just showing like what the seasonal patterns and dissolved oxygen concentrations were. And what we would expect is that when DO concentrations are higher, rates of respiration would be lower, because that's the rates of oxygen consumption are slower, and so there'd be more oxygen. And what's happening here is a little counterintuitive. What we see is that when rates of respiration are really high, when rates of oxygen uptake are really high, dissolved oxygen concentrations are also high. And then when respiration rates drop lower, so does DO concentration. So what we think is happening here is actually explained by the complex exchange between the ponds and the sloughs during a bloom event. And so to explain this pattern a little bit better, this is a zoomed in view of some of the data when there was high chlorophyll in the slough. And this is showing just a few tidal cycles. So the light gray lines are depth, and so we're using that to track the flood and the ebb tide. And the red line is the green line is Alviso, and then the red line is the pond data from the pond A8 outlet. And just going back to the map here, again, Alviso, the Alviso station is kind of like in the middle of Alviso Slough, and then the pond A8 station is right at the notch, right where pond A8 is releasing water out into, or is connected to Alviso Slough. And so what we saw in the DO and the chlorophyll is that there are these peaks that are happening during the ebb tide. So as water is dropping, there is this peak in chlorophyll and dissolved oxygen that happens first at the outlet. And then there's the same peak that's happening at Alviso. It's a very similar shape. It's just like muted. And so what's happening is that the water is passing by the pond A8 outlet. It's high in chlorophyll and high in dissolved oxygen, and then it's traveling along the slough, and then it's going past the Alviso sensor. And so that difference in dissolved oxygen is actually the respiration, the rate of respiration of that parcel of water. And what these patterns that with the tidal cycle are showing us is that it's consistent that with the hypothesis that elevated chlorophyll A concentrations are coming out of the pond, past the pond sensor, and then to Alviso Slough. And that's this being the high chlorophyll concentrations are being driven by export from the pond. And I tried to create a conceptual model of what's happening in the data. And I apologize, I'm not an artist, so this is a little embarrassing, but the best I could do to try to summarize how like these like complex concepts basically. But what we think is happening is that the phytoplankton in pond A8 is causing supersaturated DO water in the pond that is then leading to high dissolved oxygen concentrations in Alviso Slough. At the same time, this phytoplankton is also causing the delivery of biomass, this decaying phytoplankton, to Alviso Slough, which is causing high respiration rates. So we think what's happening is that there's this balance between the highly oxygenated water from the bloom and the decay or the breakdown of the biomass. And this balance can kind of tip depending on whether we're in a bloom period. So in a bloom period, there's more the high dissolved oxygen concentrations are outweighing the oxygen drawdown from the respiration. And then in non-bloom periods, you kind of see the opposite happening, that the high respiration rates in the sloughs are the rates of oxygen consumption are more rapid than the high oxygen concentration water is able to buffer. So what we think is happening is this form this really delicate balance. The interaction between the ponds and the sloughs is creating this really delicate balance. Yeah, I mean, this figure is comparing the rates of respiration measured in this study to other studies. And just to kind of illustrate the point that respiration rates are really, really high in the sloughs. Yeah, in the box, I'm showing the respiration rates in Alviso Slough in January to May. And these are exceptionally high compared to other systems, other estuaries. There's one study in San Francisco Bay that measured respiration rates kind of in a marsh on similar scales, but in general, yeah, the rates that we measured in the Alviso Slough is really high. Even in the parts of the year when the rates were a little bit lower, they're still relative to other estuaries quite high. So we think that the interaction between pond A8 and Alviso is creating potential for really rapid drops in dissolved oxygen if this balance is disrupted. In order to build on the results of Derek's study, we shifted to using a hydrodynamic model paired with in situ sensor data. So this is the other study that I mentioned that was led by Martin Valerich. So we use this model to estimate the rates of to estimate what's happening with some of these processes. And what this is showing here is a grid of a hydrodynamic model used for the tracer runs. And so essentially what Martin did in collaboration with some of our modeling collaborators is added a chlorophyll tracer to the pond and then tracked the advection of that chlorophyll or phytoplankton out into Alviso across this the Alviso cross section here, which is where our sensor is located, in order to estimate fluxes of the phytoplankton biomass out of the pond into the slough. And what he found is that okay, so this plot. This plot is comparing fluxes from the model to the chlorophyll actually measured at the pond A8 outlet. So the bars are showing mean monthly chlorophyll A flux across the Alviso cross section that I showed on the model grid, and it's divided up in the proportion of chlorophyll flux that's attributed to tidal mixing, which is the orange color, and advective flux, which is based on discharge from the Guadalupe River that feeds into Alviso. And so the combination of these two sources is the amount of chlorophyll that's fluxing past the Alviso station, and it shows that fluxes are highest during the winter and the early spring months. And the green line is from actual, it's not based on model output, it's actual data collected at the pond A8 outlet. And this is the average maximum daily chlorophyll concentration at that station. So what it's trying to capture is what chlorophyll concentrations are coming out of the pond. And we see that there's really good agreement that when chlorophyll is high in the pond, there's high chlorophyll in Alviso. And what this means is that when there's more chlorophyll in the pond, there's going to be more chlorophyll in the slough. So there, it's kind of confirming this connection between the two. And it's not a full biogeochemical model. It's based primarily on a hydrodynamic model. So it can't resolve the effect of nutrients here, but it does confirm that there's, according to the model, net transport of phytoplankton out of pond A8 and into Alviso, that the transport is driven by a combination of flow from Guadalupe River, which is connected to Alviso, and tidal pumping, and that nearly all phytoplankton that leaves pond A8 is respired within Alviso Slough before reaching the open bay. So the takeaways from these different studies are that between these different data sets, the analysis and the model output, the results support the hypothesized mechanism that that we proposed for the pond 8 complex in Alviso, that high nutrients can cause phytoplankton growth in the ponds. This is then transported to the sloughs where decomposition of phytoplankton occurs, and it can lead to low oxygen conditions. And then the extent of low oxygen conditions would depend in part on many factors, but whether there's delivery of the supersaturated water from the ponds as well. So this is a complex, this is a simple diagram, but it's a very complex process that's dependent on many different factors. And so that's just like an important caveat to keep in mind. The main, I guess, final takeaways are that the interaction between the high nutrients and the managed ponds have the potential to impact water quality in the lower South Bay sloughs, and that the delicate balance between the two create potential for low dissolved oxygen conditions. And because this system is susceptible to low oxygen, and the dissolved oxygen dynamics are tightly linked to the exchange between the ponds and the sloughs, restoration activities could potentially impact water quality, and it's something to consider during the restoration process. And there are a few, actually many remaining uncertainties and some additional work that we want to pursue. So maybe the key uncertainties that remain are the impacts of the changing wastewater nutrient loads on pond phytoplankton growth. So the dischargers in the lower South Bay are changing the way that they're processing their nutrients, and with the idea of decreasing the nutrient loads to the bay. So how will that impact phytoplankton growth in the ponds and dissolved oxygen dynamics in the slough? What effect will the pond restoration process have on water quality in the sloughs? So far, all I've been talking about really is how the conditions are linked, but what will happen as pond restoration, the restoration of these pond habitats proceeds, if there are more connections between the ponds and the sloughs. And then I guess the last point here is do the restored ponds have an ability to buffer nutrient inputs? And this is really something that I think is quite possible when they're restored to fully restored to wetlands, because wetlands do provide efficient buffers for nutrient inputs. They can a lot of denitrification can happen in them in the marshes, so there can be a lot of nitrogen removal from the system. So maybe there is or there be short-term impacts on water quality, but maybe the long-term impacts would be that they would be providing a buffer to nutrient inputs to the bay. So there's a lot of questions and uncertainty around all of that, but really interesting additional work to come from that. And our future work includes continuing to our synthesis work and modeling for the lower South Bay slough and pond data, and also to do some field data collection in the managed ponds as restoration process proceeds is one way to try to get at some of these uncertainties. And I think I'll stop there. I had a couple slides about halves, but I ended up talking longer than I expected. So I'll go to questions, but I can put this back up afterwards, because this slide has a a QR code and a link to a really cool Shiny app that Dan Killam, a scientist on our team, put together as a way to visualize a lot of the nutrient management strategies data. And so I'll go to the question slides because it's a pretty picture from one of the ponds, but I'll go back to the QR code at the end so you can check that out too.
Well, that was great. Thank you, Ari. And I see a hand up from Maya, but I also see that Kevin Lundy put a question in the chat. Kevin, do you want to read yours aloud, or would you like me to do it?
There we go. I had to get
There we
my
go.
button there. I'll read
Yeah,
it.
yeah.
Yeah. All right. Thanks. Really appreciate the presentation. You know, at the beginning, you kind of had circled nitrogen and phosphorus coming in. And so I kind of wondered, uh, is there a study that has looked at whether nitrogen or phosphorus is the bigger driver of algae growth and phytoplankton production in the sloughs? So is that kind of like well understood that one or the other is more of an important factor? Are we kind of presuming it's nitrogen because that's kind of our assumption for San Francisco Bay that that's really the driving factor? I wonder what
Yeah,
your
that's a
thoughts are on that.
good point. Yeah, the both are important nutrients for growth. The yeah, the kind of I guess the prevailing model, I guess, is that phosphorus is limiting in freshwater systems, but that nitrogen is limiting in marine systems. So usually for any study of looking at yeah, phytoplankton growth in marine systems is more focused on studying at the impacts of nitrogen because it tends to be a much stronger driver of production in marine systems, but not the case for lakes and streams and things.
Nice. Maya, go ahead.
Great. Thanks, Ari. This was so interesting. Really appreciate being able to listen. I was wondering if I think my question is a little bit related to Kevin's question, and I was a little surprised, I guess, to see the modeling results in Pond 8 and Alviso Slough where the highest chlorophyll A and understanding that was inferred was in, the winter time, so February, March, and then it's much lower, albeit still high, still in the kind of 50 to 100 microgram per liter range in the summer. But is that expected? Is that sort of the blooms are um, the biomass is higher in the wintertime? Is that a standard result for, and do we know why? Is it kind of related to nutrient availability? Is it related to the community that's out there and kind of what conditions allow them to do these big wintertime blooms?
Mm-hmm. Yeah, so yeah, are you referring to the actual data that I presented the
Well, I was looking at that modeling results slide that
oh
had
okay.
the chlorophyll trace, and then I
Uh-huh.
was trying to scroll back to see if any other slides showed. I know we saw that very large bloom in October,
Mm-hmm.
2024 in Pond 8, and to me that feels like, yeah, you get these fall blooms. Like it's still pretty warm out and the sun's shining in October. It hasn't started raining, so like blooms happen all over the place. But I
Mm-hmm.
was more surprised to see like a peak really intense chlorophyll in February and just wondering if that is a standard pattern that you see in the ponds and maybe why that would be.
Mm-hmm. Yeah, it's a really good point. In the Bay, so I would think in general, yeah, you expect more, at least in the Bay, like more of those big blooms to be happening in the spring. So not really in February, like a little bit later in the year. And yeah, in the pond yeah, the timing of the timing of the increases in chlorophyll of the phytoplankton biomass is a little, I think, unusual. I don't actually know what the driver of that seasonality may be. I and I think yeah, we have only one year of data in the ponds. So I think we would need to be collecting a little bit longer to really understand that seasonality. But the bar graphs that are from the model are that's average over several years. Martin, I think Martin may be on the call. He led the modeling analysis, and so those you can go back to them. Yeah,
Yeah,
Yeah, that one.
yeah,
Mhm.
these ones are showing the chlorophyll A is actually average over several years, right, Martin?
it is, and that's actually at the notch, the Pond A8 notch, where it goes into Albizo Slough. But in general, so we're not entirely sure why you're right about the wintertime blooms. So we just added our exos to the salt ponds themselves, but for like the last 10 years, we've had sensors in the sloughs, we do see higher chlorophyll in the winter than in the spring, summer in the sloughs pretty consistently. Um, and yeah, and so for that reason, that's something it's part of the reason why we're trying to do this modeling, right? Is try to understand that because, like I already said, we normally expect a spring bloom, or at least in the open bay, and so what
Yeah.
exactly is driving that? Hard to say, but we definitely do in general. And it varies year to year, but in general, these like late fall and winter blooms, yeah, in the sloughs, we'll see the chlorophyll spike up in Albizo and Guadalupe.
It's really interesting. And we've been looking at the algal community data from the ponds as part of this study with Valley Water, and we see just tons of picocyanobacteria. Like that's, they're just one of the dominant sort of groups out there, the tiny. Um, but there are diatoms, and so I was kind of thinking there would be a spring bloom too. So that's fascinating to me that it's really peaking in the winter. And we'll look at
Yeah,
some of we only have, I don't even think we have winter data, unfortunately. I think we only have late spring, early summer, and late summer, early fall. But maybe that's a place where folks could look in the future just to see, who's blooming there in the winter in case
yeah,
that helps explain it. Yeah.
for sure.
Thanks.
All we really have is exo data, so we don't really know what the community is. So it could be, yeah, we don't know exactly what's
Yeah.
driving
Yeah. Great. Thanks.
it.
I just wanted to call attention to Levi Lewis's comment in the chat here about the incredible macroalgal bloom in A8 in September 24. I don't know if that, like, Levi, do you want to comment on that or put that little framing around the how that ties into what Arian has presented here?
Sure. Yeah. Can you hear me okay?
Yeah. Yeah.
Yeah. Just noting that, when we look at our water quality data, we're kind of assuming that it's chlorophyll, or sorry, it's a phytoplankton that are driving a lot of the patterns. But in A8 during August, September, it was almost entirely macroalgae, and there was very little phytoplankton in the pond. I don't know how long that lasted. We only sampled for a week, but it was pretty dramatic. We couldn't even pull our trawls because every trawl was just absolutely chock full of green algae. Yeah.
Hmm. Interesting. Ellen, Jonk, thanks for being here. Do you want to ask your question aloud or shall I read it? 3, 2, 1. Ellen Jonk asks, how long in years has the criteria of 5 milligrams of DO per liter essential for fish survival been in place? And I know that's been in place for as long as I've been doing work in the Bay, but Ellen wants to confirm that's a correct question, and she's worried, or I guess wondering whether or not it's perpetually not being met, and what that might mean. And I guess related to that, I had a question about, is it only about fish and biota? Not that those don't matter, but if I'm always looking for a way to tie this to people and to communities and things like that. And so if there is a way to sort of help connect that to something beyond just the fish and the biota and how it affects people, that would be helpful too, I think.
Yeah, so the 5 milligram per liter threshold is for the open bay, not for the sloughs. Yeah, the sloughs, the water quality conditions are really dynamic and really different from the open bay, and so that threshold isn't really applicable. The work that we have done with the water board and Tetra Tech was based on the requirements, the oxygen requirements for fish that were found in the lower South Bay sloughs. And so those numbers they're calculated differently. And actually, for how long the basin plan numbers have been in place, Kevin, do you know? Sorry.
Yeah, 1975.
19, okay, well.
The original basin plan. And yeah, this is where Ari's like, it's likely not the right number, applying 5 to sloughs, but it's an option to do site-specific objectives to do that more formally. But at the moment, we're just collecting the science with all the interested parties to understand what the fish needs are relative to DO. So not getting at people, Dave, though. Appreciate that question. I guess we are focused on aquatic life. That's kind of an aquatic life-based number.
Yeah.
Yeah. Well, I just want to respond and comment because I've been looking at, as Kevin knows, and many of you here, I've been working on saving the Bay for a long time in different roles throughout my career. And I just think this is absolutely fascinating. Thank you for this work because, particularly the algae blooms, the 2022 city of San Francisco is, that's where I live. You know, what can they do to revamp their whole nutrient loading excesses and others around the Bay too? And I think, of course, this is all challenges for the water board with the backup of SFEI data. So I just think this is absolutely fascinating, and I'm just kind of looking at this trajectory. Yeah, 1975, sure. That's a long time. So anyway, I think this is important work. So thank you.
Good comment, Ellen. Thanks.
Yeah.
Dave Schulhammer, do you want to ask your question? If not, I'll read it. But Dave notes New York Slough has low DO in summer, no ponds, and no treatment plants. Maui Slough also, as I recall. There's a lot of decaying organic matter back there. Is low summer DO a natural condition in lower South Bay sloughs? And do ponds and treatment plants alter the timing and magnitude of low DO events?
Again, okay, hang on. I need to like look at
Yeah, it's a long question. Yeah.
Like, hold on 1 second.
I had so many softball questions ready for you guys.
Yeah,
But
okay.
but we're doing
I want to
technical
hear those. Okay, so yeah, I guess to this question, maybe like the first part of important context is that in habitats like sloughs and these estuarine environments, they're having big swings in dissolved oxygen is that happens without, like, even if it's not a real highly altered system. There is a lot of production in these shallow parts of estuarine systems, and then there's associated drawdown as that material decays. So that's normal. I think what we're trying to get at here is whether because of the high nutrient loads and the kind of ability for the ponds to kind of act almost as like little incubators for phytoplankton or other types of algae, as Levi pointed out, then that creates the potential for the oxygen to drop much lower than it would in an unaltered environment. We unfortunately don't have a good reference site. This has been a topic of a lot of conversation. Some of the other work I've done about a reference site in Lower South Bay. There's not really any slough we can point to that like has not been impacted. So it's a little hard to say exactly like what changes in dissolved oxygen like we would anticipate normally. But definitely in any kind of estuarine system, we would expect that to be happening to some extent. So yeah, I think that's kind of maybe to like the first part, and then to lower summer DO. Yeah, so I think, yeah, and then do ponds treatment plants alter the timing or the magnitude of low DO events? And I think I think that is what's happening in Alviso. That's why there's this kind of like change in the seasonal pattern. It's not consistent with Newark or what we would expect. And I think part of why that's happening is because the timing of the biomass production, but also then kind of the lag of that delivery of all that organic matter. It's not happening at the same place where it's degrading. Like it's being advected out into the slough where it's decomposing. And so there's like some delay that's happening with that delivery. And it takes some time for the decomposition process to occur. So the oxygen concentrations can continue to be drawn down over longer periods of time, kind of after the production or a bloom has collapsed. So I think that yeah, I think so. I think the ponds are having an impact on the timing in that sense, that because of the spatial decoupling of where the production's happening and where and the decay, yeah, it's causing the low DO events. But then also there are in summertime where we tend to see the lower oxygen concentrations. That's not just because of like respiration for material coming out of the slough. There's other organic material in the system, and then there's also lower oxygen, higher temperatures. Like the warmer periods of time, or when oxygen concentrations tend to drop lower as well. So yeah, sorry, it was kind of like a long answer. Hopefully
right.
that
Well,
answered your question.
we're nearing the end, but I do see Jim Mervin has his hand up, and I am not going to get in Jim's way. So, Jim, go ahead and ask your question, and then we'll wrap it up.
It's a little bit of a question and some observation. Yeah, we were, I ride along with the UC Davis, the Lewis's team, the UC Davis crew that's on the Otolith Geochemistry Fish Laboratory, Fish Ecology Laboratory. And so, doing monitoring of Lower South Bay and picking up the fish. Yeah, some years ago, when Pond A18, which is owned by City of San Jose, but it was open to circulation in 2006, and then Pond A16, which is a managed pond, was also opened about the same time. I noticed for some years initially, the water was like emerald green that would discharge from those ponds in the summer. Well, maybe it was spring. I took many photos of it. It was like the most unnatural green color you've ever seen. It like glowed green. But that died down over time, and this is part of the colonization of the benthic organisms like the mud snails and such. They get in the pond and they start chewing down on that chlorophyll. But it was very visible. This was a system upset when you took a hypersaline pond and all of a sudden started circulating it with the bay. And yeah, it's interesting that A8 seems to be still cycling through that process, even though it's been many years. I guess 2013 is what I saw from your slide, that when it was first initially breached. So that colonization or that regularization hasn't really happened, and it might be because the pond is so big and the flow is kind of restricted. They see it. The fish folks see that also in the type of fishes that they catch. They catch a lot of silver sides, a lot of Shimafuri gobies, many more than we see in other parts of our trawling surveys in summertime. So numerous indications that you're not really dealing with well, as I think it's really not functioning as a natural system yet. On the other hand, Pond A8, Pond A21, and Pond A19, which have been restored to natural restoration, they've vegetated over, and we're getting a lot of our counts of longfin smelt and anchovies are generally the highest, except for last month. That's a different story, but are generally the highest in those restored ponds. So anyway, there's an evolutionary process. The only other comment I'd make is that we catch anchovies in the upstream sloughs in Lower South Bay in the summertime very often when dissolved oxygen dips below 2, if you can believe that. When supposedly fish don't even survive, we catch lots of anchovies. And apparently, well, they're there for spawning. They're unimpeded by that low DO. So the marsh system, well, it accommodates many of the native fish. In fact, the low DO becomes a barrier to non-native fish, which is interesting. And I'll leave it at that. It's just a bunch of random.
All right.
Oh boy.
I love that discussion. I love all
Yeah.
of that. I wish we had another half hour, but I have a meeting at 12:15 that I need to get to. So we're gonna let everyone off the hook. All right, I'll save the softballs for another time. But can DO ever be too high?
I mean, it gets super saturated, but then it doesn't like it then kind of equilibrates with the atmosphere. So
not really.
no, it doesn't. I don't think it lasts that long.
See,
I don't know
those are
if
the
like
questions I have for you.
physiologically if it could have
Yeah.
any negative effect, but like some of the fish people on the call would be able to answer that better than me.
All right. Well, thank you everyone for coming. Thank you for this great presentation and discussion. Have a wonderful afternoon, and look on the website for the recording soon if you need it.
Thank you so much. Thanks everyone
Thanks,
for
everyone.
joining.